Hi, my name is Pitt and I'm studying local selection and landscape genomics in livestock species at Cardiff University. And what I want to talk about today is a group of animals I find particularly interesting, Chillingham cattle. So Chillingham are a unique breed of cattle situated at the Chillingham Park private estate in northern England. The herd here are feral, they're essentially wild and remain mostly unmanaged, with only rare veterinary intervention and the occasional supplementary feeding. Records indicate that this herd has been isolated since the 14th century and has remained totally isolated from any other cattle in the past 300 to 350 years. The herd provides an interesting insight into ancient and wild cattle. Males compete for mates, establish home ranges, and individuals fill the ecological niche of a megaherbivore. Unusually for mammals, the breed also exhibits aseasonality, so mating all year round. And over the last few centuries, the population size has stochastically fluctuated, with a particularly severe bottleneck down to 13 individuals in the 1940s. And as of 2019, there were 108 individuals, while the estate has an overall estimated carrying capacity of 120. Considering demographic history and isolation, it was expected this breed has experienced high levels of inbreeding, yet phenotypically has displayed minimal loss of fitness. And I think I will leave the next point to the official website where they say, as no new animals have joined the herd for the last 300 years, one of the big scientific questions is how do they cope with all this inbreeding? You could argue that scientifically they cannot exist. So this of course brings us to our hypothesis, do they exist? No, but while this website finds an odd way to word it, there is a good underlying point. While we commonly see high inbreeding in cattle, it's usually a result of targeted artificial selection. However, with the lack of human intervention, the situation in Chillingham is far closer to a wild population. In such examples, the frequent mating of related individuals lead to inbreeding depression, where there is increased levels of homozygosity, and therefore frequent expression of recessive deleterious alleles. This often results in reduced fitness. A few of these examples include Channel Island foxes, Scandinavian grey wolves, and the banded mongoose. Populations like these often experience high levels of inbreeding as a result of limited population size. However, unlike Chillingham, they still often receive migration from neighbouring populations. And as far as I've looked, I've not come across a wild system that is as entirely closed as Chillingham, although I'd love to hear about any if you've got any. So a few previous studies have looked into Chillingham so far to identify levels of inbreeding and homozygosity. Microsatellite markers were first used, where 24 out of 25 were completely homozygous. This was followed by 8 individual samples of mitochondrial DNA, all carrying the same haplotype. And then finally, a high-density SNP array with over 700,000 markers, where it was estimated that only 2.6% of the genome was heterozygous. To put that into perspective, a highly inbred commercial cattle breed tends to have heterozygosity at least 10-fold higher than that. These are all great indications that point to a highly inbred herd with unusually high levels of homozygosity. However, all these data are vulnerable to ascertainment bias because the markers that had been defined were originally defined in populations quite distantly related to Chillingham. So it means that when it's applied to Chillingham, you may be severely underestimating the variation. You're just completely missing them. So what we did was some whole genome resequencing 10 Chillingham individuals to try and capture all the variation within the population. Throughout the study, although not a perfect comparison, we used 10 Angus individuals, which was the most closely related breed available for the 1,000 Bulls Genome Project, to provide some sort of baseline context. So general variation first, we identified 3.5 million biallelic SNPs that pass quality control in the data set, with only 367,000 variable in Chillingham, but also identified 12,000 unique variants within the breed. And without throwing too many dry numbers forward, I can basically summarize in saying that the variation in Chillingham, as with the previous SNP study showed, is around 10 times lower than comparative breeds. For instance, heterozygosity per individual was 10 times lower in Chillingham, general measures for compared to Angus, sorry. And general measures for the species Bossaurus tend to range between 0.3 and 0.6, which is much higher than what we see in our data. This basically means if you take one random individual chilling in, you don't expect to see a heterozygous site once every 20,000 base pairs, which is quite insane. So as the majority of Chillingham's genome seems to be without variation, we first looked at runs of homozygosity, and as the name suggests, runs of homozygosity are continuous segments of the genome that are predominantly homozygous. This can assist with identifying levels of inbreeding, as runs of homozygosity are often analogous to autologous regions. Typically, over generations, autologous regions are shuffled by recombination, therefore the presence of the longer runs are indicative of more recent or higher levels of inbreeding. So this plot that I've got here is a run of homozygosity plot, and it's just for chromosome 1. Each horizontal level is a different individual, so 10 Angus individuals at the top and 10 Chillingham below. The coloured segments show where a run is. So a couple of things that might stand out here from the perspective of Chillingham is that the runs span almost the whole chromosome if you added them together. The runs are longer in general, and the runs are more consistent between individuals of the same population within Chillingham. This is observed across the entire genome and reaches to such an extent on chromosome 28 that effectively a single run covers the whole genome, the whole chromosome, sorry. Calculating runs derived from inbreeding coefficients as a proportion of the genome covered by runs of homozygosity gave coefficient estimates of around 0.91 for Chillingham compared to just 0.13 in Angus, so it really shows the difference. These results together indicate that there's extensive and prolonged inbreeding still occurring in the recent past, despite the recovering numbers of the population. However, there seems to also be consistent gaps between runs of homozygosity, which are shared between, within the population, perhaps indicating some sort of isolated pockets of variation. To find the extent and potential structure of the variability, we measured heterozygosity in sliding windows spanning 100 kilobases. We identified windows that were consistently high across each breed, giving us heterozygosity peak windows. Again here, the plot, Angus at the top and Chillingham below. The coloured peaks show windowed heterozygosity averages across the genome, and the grey indicates, it marks heterozygosity peak windows. So although there seems to be far more peak windows in Angus, more of the grey lines, it's just the way that the plot's done. The number is actually quite similar, so there's around 10,000 and 11,000 for Angus and Chillingham respectively. However, there is considerable more overlap clustering within Chillingham, which produces a far from uniform or random distribution. Additionally, there is a massive surplus of loci that have an excess of heterozygosity, deviating from Hardy-Weinberg expectations. So a quarter of Chillingham sites as heterozygosity peak windows had an excess heterozygosity, which is about 2.6 times higher than the genome averages, which could be an indication for the presence of balancing selection across these regions. So interestingly, heterozygosity peak windows are host to a higher ratio of non-synonymous to synonymous mutations in Chillingham, which means that half the polymorphic exonic loci resulted in a predicted amino acid alteration. SIFT scores are a metric that uses phylogenetic frequencies of a mutation, i.e. how many times a mutation has occurred across multiple species, to predict the probability of the tolerance of that mutation. So Chillingham also displayed higher levels of deleterious variance relative to the remainder of their own rest of the genome, and also to Angus. This could potentially indicate that there is cost, or showing the cost that's associated with balancing selection, as deleterious alleles are also increased in frequency, or perhaps indicating that purging selection is ineffective in such regions. So for functional effect, fortunately cattle have an extensive resource available that allow insight into the further functional effects of the genomic architecture. We look primarily at quantitative trait loci, or QTLs. These are specific genomic regions that have a measurable phenotypic effect on the individual. In the case of cattle, it's quite good quality because they're often narrowed down to a specific mutation rather than a broad region. We screened the location and enrichment of 100,000 QTLs to test if particular traits or functions were specifically associated with these heterozygosity peak windows in Chillingham. We found that 13 out of the 390 traits that we tested were significantly rich and most heavily associated with fertility. We found it surprising that fertility and milk production, as you can see, were the only two categories that jumped out as significant, as we expect immunity to be highly prominent, considering the devastating effect that pathogens and disease can have on small inbred populations, and also the observation that Chillingham doesn't really seem to have any of these issues. To investigate further, we looked at a specific region, the bovine MHC. So this was of particular interest because not only is it heavily involved in immunity, it's also highly conserved and one of the most variable regions across vertebrates. In cattle, the MHC is isolated to chromosome 23 and is comprised of 3 classes. So these 3 plots here show class 2 and 3 of the bovine MHC. And the top half of the plots are the runs of homozygosity, same as before. And this is just showing Chillingham. Then the bottom halves show the excess, the significant excess of heterozygosity, where the height of the points indicate more of an excess. Red segments, and the red segments are showing key immunity-related genes. So overall, the main thing from this is just the sheer lack of variability across most of the MHC. 2B, for instance, in the top right, showing no variability and the odd variable SNP scattered throughout the rest of the regions. Runs of homozygosity are uniform across all the regions pretty much, whereas they're almost ubiquitously high in other vertebrates. To put this into a little bit of perspective, this is the same region, this is 2B as well in Angus, and it's populated by mostly highly variable SNPs and also next to no runs of homozygosity. And then finally, this is the class 1 region, the only class where there was actually a substantial cluster of heterozygosity in Chillingham. This was surrounding the gene BOLA-NC1, which has been shown to play a role in pathogen subversion and also acts as an immunosuppressive factor. These two functions are carried out by different isoforms, so it's possible that the expression of both isoforms may only be possible through maintenance of variability in this region. So in conclusion, Chillingham provide an excellent example of the viability of conserving a locally adapted breed in a semi-wild habitat, despite severe bottlenecks and minimal management. Also the identification of regions and the abundance and the scarcity of variation seen across different regions provides insights into the evolutionary history and the selection pressures applied to small inbred mammalian populations. And notably, balancing selection and high heterozygosity does not seem to center around regions involved in immunity, but instead associated with fertility. The notable lack provides further questions into the historical pathogen burden of the breed and also the adaptive potential to respond to emerging diseases in the future. Some possible next steps, I'd quite like to look at copy number variants, so taking some longer sequence reads to see if some of the variation unique to Chillingham is hidden there. Also epigenetic markers, some of the same thing to see if that's modulating some of the expression. And also simulating drift, purging, and balancing selection to understand the presence and strength of these processes that are occurring here. So thank you for listening to my talk, and my future self can hopefully answer some of the questions now.